Biology in Motivirappa Government PU College, Davangere. So today's topic we are going to deal is biotechnology and its application. You all know about biotechnology. So biotechnology is nothing but the manipulation of the genetic material or a genome of any organisms to obtain a desired product. And the products may be biologicals or biopharmaceuticals. So we are going to deal with the biotechnology and its application. Let us see what biotechnology deals with. What biotechnology deals with. Biotechnology deals with the large industrial scale production of biopharmaceuticals biopharmaceuticals as well as biologicals biologicals from genetically modified organisms from genetically modified organisms. So what are these biopharmaceuticals and biologicals? They are all the products that is very useful for the human welfare. That involves antibiotics, vaccines, medicines, and various things. Let us see what are the applications of biotechnology. The applications of biotechnology, that means where we can use this biotechnology. You know that biotechnology is also called RDNA technology or genetic engineering. So where these techniques are used, in which field these techniques are used. So in first and foremost, the uses and applications of biotechnology are in the field of therapeutics. Next, in the field of diagnostics. Next, in the field of genetically modified crops in agriculture. Crops in agriculture. And the next, that is, it is used in gene therapy. And this technique is also used in the processed food. And it is also used in the waste management. That is also called the waste remediation. It is also used in water treatment. And also used in fuel productions. That means energy productions. Energy, biofuel productions. Energy means here biofuel productions. So these are the uses or applications of biotechnology where the techniques of the biotechnology or RDNA technology is used in these all areas. So let us see what are the three critical areas that are used in biotechnology. So first of all, that is during the biotechnology, in the techniques, they concentrate on providing a best catalyst. And that best catalyst may be in the form of a microbe or an enzyme that is able to use further for cloning the genes. And the next one, for cloning the genes, we need optimum conditions. And the conditions will require a setting up of a proper pH, proper nutrient medium, and proper like artificial environment condition inside the laboratory or inside the bioreactor. That is the second critical research area. And the third research area, after obtaining all these products in the bioreactor, we have to process it. That is a downstream processing techniques. And these downstream processing techniques is to purify the product from the bioreactor. So these three are the critical research area that is used in biotechnology, providing the best catalyst in the form of an improved organisms. It may be a microbe or an enzyme, and there one is the creating a sterile environment or a optimum conditions for a catalyst to act, and another one is the third one, that is the downstream processing techniques for the purification of the products. Now let us see the applications of biotechnology in the field of agriculture. Applications of biotechnology in agriculture. So there are three options. You can go with any of the options. If you want to go with the agriculture, if you have a main profession as an agriculturist, then you can opt all these three areas. One, you can go with the agrochemical based agriculture. That is the very oldest method of agriculture, agrochemical based agriculture. And the next one, that is the recent one, that is an organic agriculture. And third and the very recent one, that is a genetically modified crop based agriculture. So you have heard about the green revolution. So the green revolution was the happening in the agriculture when Norman Borlaug, he thought that the world is under the scarcity of the food and there was not sufficient food supply to the people around the world. So he thought that to improve the crop varieties and also increase the yield. So he decided to do something about the agriculture, and he started the green revolution, where he main concentrated on wheat and paddy and other main grains. And the same, it was followed by Indians also. The Indian scientist, this M.S. Swaminathan, also followed it, took training under the Norman Borlaug. Norman Borlaug is the father of international green revolution, whereas M.S. Swaminathan is the father of Indian green revolution. So they mainly concentrated on the agrochemical based agriculture, where they used the agrochemicals. So what are agrochemicals? Agrochemicals are the chemical fertilizers and chemical pesticides, insecticides, bactericides. All these chemical substances are used to prevent diseases and to prevent crop loss by the pests and diseases, and to improve the yield, to increase the yield. So agrochemicals are the main the things that were mainly used in agrochemical based agriculture. And then later what happens, the organic agriculture came because of the side effects of the pesticides and insecticides and other things, that they thought that natural way of agriculture is better. That's why then the organic agriculture was started. And later on in the recent era, all these two agricultural methods where the conventional, they are related to the conventional agricultural method, that is a traditional agricultural method. So they thought that we need, the recent scientists, the biotechnology, they thought that the agriculture will be yield better and very effective and fruitful only when we use the genetically based, genetically modified crops. Okay, that is one. And what are the impact of green revolution? Impact of green revolution is they increased they tripled the food supply, and they also increased the yields, and they also used water. The farmers, they started use of better agricultural practices, like they used machines for tilling, ploughing, harvesting, etc. And they also used the better irrigation methods also, like drip irrigation and all. And also they used much agrochemical based. Though the what are the impacts? Impacts are the increased yield and improved crop varieties, and the use of better agricultural practices, and the use of what agrochemicals. But still, this was not enough to supply food for the people around the world. So nowadays, because of the shortage of the land, and the more and more diseases and pests, and so many environmental what disasters are happening, that's why the scientists recently, they are going with the genetically modified crops. So now, and genetically modified crops, when it comes in the mind, so before that, there is a one step back. If we go, we have all heard about the tissue culture. We have all heard about the tissue culture. So in a tissue culture, we take a cell or a tissue or any part of the plants as explants, and we grow it in a test tube. That means it is an in vitro cultivation, in vitro culturing of the plants, parts of the plants, in a sterile condition, in a special nutrient medium. So this tissue culture is one of the best method, because we can grow much more. That means large quantities of plants in a short period of time. So this here, the plants they are obtained are all identical to each other, because we use only cell or we use tissues. So they are all somatic. Those all the plants which are produced from the what somatic tissues or any cells, they will be similar in genetically as well as morphologically. So all these genetically and morphologically similar plants, they were obtained from the tissue culture techniques. They are all called soma clones. They are called soma clones. Soma clones. So we here in a tissue culture, we take explants. So what are explants? Explants are part of the plant that we taken out and culture in a test tube, in a sterile condition, in a special nutrient medium. And these special nutrient medium must contain the substances that stimulates the growth of the plant. Like it should contain what the starch and nutrient like organic, inorganic substances. Starch is organic substances. We need sucrose little bit, and also the hormones, auxins. They are all required for the preparation of the nutrient medium. And any cells that we take that has a capacity. That means the cells or the tip of the stem or the root that contains meristems, they are all undergoing uncontrolled cell division, and it is having the capacity to develop into a whole plant. ಒಂದು ಸಣ್ಣ ಕೋಶ ಅಥವಾ ಯಾವುದೇ ಒಂದು ಮೆರಿಸ್ಟೆಮ್ಸ್ ಗಳನ್ನ ಅಥವಾ ಎನಿ ಪಾರ್ಟ್ ಆಫ್ ದಿ ಸೆಲ್ಸ್ ಪ್ಲಾಂಟ್ಸ್ ಅನ್ನ ತಗೊಂಡ್ರೆ ಅದಕ್ಕೆ ಒಂದು ಸಾಮರ್ಥ್ಯ ಇರುತ್ತೆ. ಅದೊಂದು ಪರಿಪೂರ್ಣವಾದಂತ ಒಂದು ಸಸ್ಯ ಆಗುವಂತ ಒಂದು ಸಾಮರ್ಥ್ಯವನ್ನ ಹೊಂದಿರುತ್ತೆ. ಸೊ ಈ ಸಾಮರ್ಥ್ಯಕ್ಕೆ ಏನ್ ಹೇಳ್ತಾರೆ, 2T potency. This capacity is called 2T potency. So the capacity to grow a whole plant by any cell or explant is called what? Is called what? 2T potency. 2T potency. And by using this principle, the explants are grown in a tissue culture, that is an in vitro culture. Tissue culture means it is a in vitro culture. And we grow thousands of plants. We grow thousands of identical plants, and this method is called micropropagation, and this method is called micropropagation. Micropropagation. ಸೊ ಟಿಶ್ಯೂ ಕಲ್ಚರ್ ಅದನ್ನ ಅಂಗಾಂಶ ಕೃಷಿ ಅಂತ ಹೇಳ್ತಾರೆ ಅಥವಾ ಕೋಶದ ಕೃಷಿ ಅಂತ ಹೇಳ್ತಾರೆ. ಸೊ ಇಲ್ಲಿ ಒಂದು ಕೋಶ ಅಥವಾ ಅಂಗಾಂಶ ಅಥವಾ ಸಸ್ಯದ ಯಾವ್ದು ಆಂಥರನೋ ಇರ್ಬೋದು ಅಥವಾ ಪೋಲನ್ ಇರ್ಬೋದು, ಹಾಗೇನೆ ಅವುಗಳಲ್ಲಿರುವಂತಹ ಯಾವುದೇ ರೂಟ್ ಟಿಪ್ ಯಾವ್ದಾದ್ರು ಒಂದು ಸೆಲ್ ಅನ್ನು ತೆಗೆದುಕೊಂಡು ಅದರಲ್ಲಿ ನಾವು ಒಂದೇ ತರದ ಅಂದ್ರೆ ಒಂದು ಇನ್ನೊಂದನ್ನು ಹೋಲುವಂತಹ ಸಸ್ಯಗಳನ್ನ ನಾವು ಲಾರ್ಜ್ ಸ್ಕೇಲ್ ನಲ್ಲಿ ತುಂಬಾ ಬೃಹತ್ ಪ್ರಮಾಣದಲ್ಲಿ ಪ್ರೊಡ್ಯೂಸ್ ಮಾಡುವಂತ ಒಂದು ತಂತ್ರಜ್ಞಾನ, ಅದನ್ನ ಏನ್ ಹೇಳ್ತೀವಿ, ದಟ್ ವಿ ಕಾಲ್ ಇಟ್ ಆಸ್ ಅ ಮೈಕ್ರೋ ಪ್ರೊಪಗೇಷನ್. ಸೊ ಇನ್ ದಿಸ್ ಮೈಕ್ರೋ ಪ್ರೊಪಗೇಷನ್ we can also fuse two different cells. We can also fuse two different, if you want a plant with two different characteristics, or if you want a plant with two different products, then what you have to do is to take one cell from the one plant that you are desiring and take another cell from the another plant, remove the cell wall. When a plant cell, the cell wall is removed, it becomes what? Protoplast. And these two different protoplast, you fuse it and you water culture it in a test tube under sterile condition in a special nutrient medium, and you get a whole plant with the two different characters. That technique is called somatic hybridization. That technique is called somatic hybridization. Somatic hybridization. So what is somatic hybridization? This is the method of fusion of two different protoplast. Two different protoplast taken from two different plants with the desired characters, and you obtain one plant with the two product. So here one experiment has done and developed one plant that is called pomato. That is called pomato. So the example is pomato, where the protoplast from the potato and the protoplast from the tomato plant are fused, and they developed one new plant that is what we call it as a pomato. But it is not successfully what happened in all over the world. That is mainly used in certain countries, highly developed countries like Singapore and that Japan and all other countries. That is about the somatic hybridization. Now, if you want to grow virus-free plants, what you are using as an explant? So here you are using the meristems. And if you want to obtain a virus-free plants, you have to go with the meristems. So meristems, you know, they have the group of cells which are undergoing continuous cell division, and they have not yet differentiated into a permanent cells. So the meristem culture, it is called meristem culture. It's also a part of the micropropagation. It's also a type of the tissue culture where meristems are mainly used to produce virus-free plants. So now you know that what are the importance of the tissue culture. One is you can produce large number of plants at a short period of time through micropropagation. Another one, you can produce a soma clone, somatic hybrids. These are the somatic hybrids. You can also produce somatic hybrids, and you can also produce water virus-free plants. So this is the use of what tissue culture technique. Now, genetically modified crop, the recent one that is very much used in agriculture, that is the genetically modified crops. Genetically modified crops. What are genetically modified crops or GM crops? So genetically modified crops are those crops whose genes have been altered, manipulated by using techniques, biotechnology. So there are crops They are meant for why these GM crops are used? Because they are what are the uses of genetically modified crops? Because they are more tolerant to the abiotic stress. They are more tolerant to abiotic stresses. So, what are abiotic stresses? These are environmental stresses. Environmental stresses like drought or flood or infertility of soil, water scarcity. So, all these are environmental stresses. Still, they can survive. Still, they can survive. GMO crops can survive. And the next one, that is, they help to reduce. They help to reduce the reliance on agrochemicals or chemical substances, chemical fertilizers and pesticides and pesticides. They don't need any chemical fertilizers or pesticides. They can grow. So that means reliance means these are do not these plants do not depend on the chemical fertilizers or chemical pesticides or insecticides. ಇದಕ್ಕೆ ಕೀಟನಾಶಕಗಳ ಅವಶ್ಯಕತೆನೂ ಇಲ್ಲ. ನೀರು ಕಡಿಮೆ ಇದ್ರೂ ಬೆಳೆಯುತ್ತವೆ. ಯಾವುದೇ ಎನ್ವಿರಾನ್ಮೆಂಟಲ್ ಕಂಡೀಷನ್ ಹೇಗೇನೇ ಇರಲಿ, they can survive. And then they also helped in reducing post-harvest loss. Post-harvest losses. That means the yield, the products, if grain, food products, any food cereal products, pulses or fruits or vegetables, they have a more lifespan. They do not perish very easily. That means during the harvest, these crops won't get damaged. So that's why it also reduces water, the post-harvest losses. And these crops are more efficient in mineral uptake. In mineral uptake. Thereby, they also water maintain the soil fertile. Even if there is a shortage of minerals also, whatever available in the soil, they can efficiently absorb it. So they have such a capacity. And then enhanced nutritional value. Enhanced nutritional value. That means these genetically modified crops can be modified in such a way that even though there is no certain vitamins are there, you can also make them to produce the required vitamins also. For example, the golden rice. Golden rice is yellow in color. It is rich in vitamin A. It is rich in vitamin A, where vitamin A is in the form of beta carotene, not a easily absorbable vitamin A. It is in the precursor form, that is the beta carotene, and beta carotene, when you ingest, it is converted into what vitamin A. So these are what the uses of what genetically modified crops. I hope you understand. ಸೊ ಜೆನೆಟಿಕಲಿ ಮಾಡಿಫೈಡ್ ಕ್ರಾಪ್ಸ್ ಇಂದ ಏನೆಲ್ಲ ಪ್ರಯೋಜನ ಇದೆ? ಒಂದು ಯಾವುದೇ ಒಂದು ವಾತಾವರಣದಲ್ಲಿರುವಂತಹ ಸ್ಟ್ರೆಸ್ ಕಂಡೀಷನ್ ಅನ್ನ ಅದು ತಡೆಗಟ್ಟಿ ಬದುಕುಳಿಯುತ್ತೆ. ಇನ್ನೊಂದು, ಅದು ಕೆಮಿಕಲ್ ಯಾವುದೇ ಒಂದು ರಾಸಾಯನಿಕ ವಸ್ತುಗಳ ಮೇಲೆ ಅವುಗಳ ಏನು ಇಳುವರಿ ಡಿಪೆಂಡ್ ಆಗಿರಲ್ಲ. ಆ ನಂತರ ಅದು ಹಾರ್ವೆಸ್ಟ್ ಒಂದ್ ವೇಳೆ ಅವುಗಳನ್ನೆಲ್ಲ ಹೀಗೆ ಇಳುವರಿ ಬಂದ ಮೇಲೆ ಅದನ್ನೇನ್ ಮಾಡ್ಬೇಕು, ಅದನ್ನ ಕಲೆಕ್ಟ್ ಮಾಡ್ಬೇಕು. ಸೊ ಆ ಟೈಮ್ ನಲ್ಲಿ ಲಾಸ್ ಆಗೋದು, ನಷ್ಟ ಆಗೋದನ್ನ ಇದು ತಪ್ಪಿಸುತ್ತೆ. ಹಾಗೇನೆ ಇದು ಹೆಚ್ಚು ಹೆಚ್ಚು ಮಿನರಲ್ಸ್ ಗಳನ್ನ ಮಣ್ಣಿನಿಂದ ಹೀರ್ಕೊಳ್ಳುವಂತ ಸಾಮರ್ಥ್ಯವನ್ನು ಹೊಂದಿದೆ. ಹಾಗೇನೆ ಇದರಲ್ಲಿ ಕೂಡ ನ್ಯೂಟ್ರಿಷನಲ್ ವ್ಯಾಲ್ಯೂಸ್ಗಳನ್ನು ನಾವು ಜಾಸ್ತಿ ಮಾಡಬಹುದು. ಅದನ್ನ ಜೀನ್ ಮ್ಯಾನಿಪುಲೇಟ್ ಮಾಡಿ ಅದರಲ್ಲಿ ವಿಟಮಿನ್ಸ್ ಇಲ್ಲದಿದ್ರು ಕೂಡ ಅದು ವಿಟಮಿನ್ಸ್ ಪ್ರೊಡ್ಯೂಸ್ ಆಗುವಾಗೆ ಕೂಡ ಮಾಡಬಹುದು. ಸೊ ದೀಸ್ ಆರ್ ದಿ ಯೂಸಸ್ ಆಫ್ ಜೆನೆಟಿಕಲಿ modified crops. So now let us see what are the genetically modified crops nowadays that is widely used. So example that is the pest resistant crops, pest resistant crops. Example that is BT crops. These BT crops ನಲ್ಲಿ ಯಾವುದೆಲ್ಲ ಬರುತ್ತೆ? BT ಕಾಟನ್ ಬರುತ್ತೆ, BT ಕಾರ್ನ್ ಇದೆ, BT ಸೋಯಾಬೀನ್ ಇದೆ, BT ಪೊಟ್ಯಾಟೊ, BT rice, etc. etc. So all these crops are genetically modified crops, but they have the resistant to the pest like insects, insect pests. So here BT cotton is a major example. That is a major commercial crop. That is why, and it is mostly damaged by three groups of insects: Lepidoptera, Diptera, and Coleoptera. So the major pests that harms all these crops comes under three groups. So these are the Dipteras, so these are the flies, and these are the bollworms. Bollworms. See, you see that the bud, the cotton, twig that is eaten by the larva of the bollworm. So bollworm caterpillar, and these are all the bollworms. See, the caterpillar of the bollworm is eating the tobacco, cotton leaves. So these are all the insects that harms the cotton plants. Here the BT crops are made pest resistant. The BT crops are made pest resistant by using the genes of Bacillus thuringiensis. Bacillus thuringiensis. This Bacillus thuringiensis in short form, in abbreviated form, it is named as BT. BT means here Bacillus thuringiensis. And here the three genes from the Bacillus thuringiensis are taken and it is introduced into the plant cells, particularly all these BT crops, and they are made resistant to all these pests. Okay, bollworms and budworms, armyworms. There are so many insects are there. So lepidopterans are mainly involving tobacco budworm, armyworms. Coleopterans mainly involves beetles and weevils also, and dipterans mainly involves flies and mosquitoes. So here the three genes, which are the three genes that was taken from the Bacillus thuringiensis and inserted into the cotton plant. And these three genes are the CRY1, the CRY1AC, the CRY2AB, and the CRY1AB. The CRY1AB, the CRY2AB, and CRY1AC. CRY means what? CRY is for a crystal. These genes, they are able to produce a prototoxin, an insecticidal toxin. And these toxins are secreted in the form of crystals. They are secreted in the form of crystals. So they are called crystal proteins also. Crystal toxins, crystal proteins, or insecticidal proteins, or insecticidal toxins. And these genes are incorporated into the plant body using a host cloning, that is using the agrobacterium tumefaciens, sorry, using the cloning vector agrobacterium tumefaciens, they are made in such a way that these what genes, they secrete these crystal proteins, and when it was ingest these any part of the plant, this bt, the leaves of or any part of the bt plants, when the larvae of these insects, they eat those leaves or any fruit or anything, these crystal proteins, they enter into the gut of the larvae. And there the crystal proteins, because of the pH, alkaline pH inside the gut of the larvae, caterpillar, and these crystals dissolve and they become liquid. And this increases the pores, this enlarges the pores. They increases the size of the pores and they enlarges the pores. So these fluids enter into the epithelial cells of the gut. And because of the over diffusion of this liquid into the cell burst. And due to the internal damage, the insects dies. So like this, the pests are prevented from eating the parts of the plants. This is why the three genes are used. So these three genes, particularly the Cry1A and Cry2AB, that controls the cotton bollworm, and Cry1AC that controls the corn borer. Corn borer. Okay. So you all know. So BT, that means it represents Bacillus thuringiensis. Three genes taken from the Bacillus thuringiensis and is incorporated into the cotton plants using the what? Cloning vector, that is agrobacterium tumefaciens, and they are these three genes, they code for the crystal protein, insecticidal proteins, and these proteins, when it is, that means when these proteins are ingested by the larvae of any of these insects, they dissolve due to the alkaline pH of the gut, and the epithelial cells, what they absorb more and more this crystal protein, and the cells burst. Like that, the larva dies. So this is about the BT cotton. Next, another pest resistant crop, that is the tobacco. You know that tobacco is the most important commercial crop. It is widely used all over the world. And it is most infected by a nematode, a roundworm, a teeny nematode. It causes infestation into the root of the tobacco plants, and they grow inside the root. So that the root, what it becomes ನಾಟ್ ಲೈಕ್, ಇಟ್ ಬಿಕಮ್ಸ್ ನಾಟ್ ಲೈಕ್, ಗಂಟು ಆಗುತ್ತೆ. ಆ ಹುಳ ಏನಾಗುತ್ತೆ, ಅದರ ಬೇರಿನ ಒಳಗಡೆ ಯಾವುದು ಹುಳ, ಮೆಲೈಡೆ ಇನ್ಕಾಗ್ನಿಷಿಯಾ ಎನ್ನುವಂತ ಒಂದು ದುಂಡು ಹುಳ, ಒಂದು ರೌಂಡ್ ವರ್ಮ್, ಅದು ಅದರ ಮೊಟ್ಟೆ ಹೊಡೆದ ತಕ್ಷಣನೇ ಆ ಹುಳ ಆ ಬೇರಿನ ಒಳಗಡೆ ಹೋಗಿ ಸೇರ್ಕೊಳ್ಳುತ್ತೆ. ಅದರಲ್ಲಿ ಅದು ಬೆಳೀತಾ ಹೋಗುತ್ತೆ, ಅದ್ರ ನ್ಯೂಟ್ರಿಷನ್ ಅಂತ ತಿನ್ಕೊಂಡು ಆವಾಗ ಅದು ಗಂಟು ಆಗುತ್ತೆ. ಆ ಗಂಟು ಆಗುವಾಗ ಅದಕ್ಕೊಂದು ಕಾಯಿಲೆ ಬರುತ್ತೆ. ಯಾವುದಕ್ಕೆ? ತಂಬಾಕು, ಟೊಬ್ಯಾಕೊ ಗಿಡಕ್ಕೆ ಒಂದು ಕಾಯಿಲೆ ಬರುತ್ತೆ. ಅದರ ಲೀಫಿನ ಕ್ವಾಲಿಟಿ, ಅದ್ರಲ್ಲಿ ಮೈನ್ ಕಮರ್ಷಿಯಲ್ ಯಾಕೆ ಟೊಬ್ಯಾಕೋನ ಬೆಳೆಸ್ತಾರಂತಂದ್ರೆ ಅದ್ರ ಎಲೆಗಳಿಗೆ, ತಂಬಾಕಿನ ಎಲೆಗಳಿಗೆ. ಹಾಗಾಗಿ ಅದ್ರ ಕ್ವಾಲಿಟಿ ಕಡಿಮೆ ಆಗುತ್ತೆ. ಆ ಕಾಯಿಲೆಗೆ ಏನ್ ಹೇಳ್ತಾರೆ? ರೂಟ್ ನಾಟ್ ಡಿಸೀಸ್ ಅಂತ ಹೇಳ್ತಾರೆ. ಆ ರೂಟ್ ನಾಟ್ ಡಿಸೀಸಸ್ ಅನ್ನ ಪ್ರಿವೆಂಟ್ ಮಾಡ್ಲಿಕ್ಕೆ, ಹಾಗೇನೆ ಮೆಲೈಡೆ ಇನ್ಕಾಗ್ನಿಷಿಯಾ, ಈ ನಿಮ್ಯಾಟೋಡ್ ಅದರ ರೂಟಿನೊಳಗಡೆ ಬೆಳೆಯಬಾರದು ಎನ್ನುವ ಕಾರಣಕ್ಕಾಗಿ ಅಲ್ಲಿ ಏನ್ ಮಾಡ್ತಾರೆ, ಒಂದು ಕಾನ್ಸೆಪ್ಟ್ ಅನ್ನ ಬಳಸ್ತಾರೆ. ಆ ಕಾನ್ಸೆಪ್ಟಿಗೆ RNA ಇಂಟರ್ಫೆರೆನ್ಸ್ ಅಂತ ಹೇಳ್ತಾರೆ. RNA ಇಂಟರ್ಫೆರೆನ್ಸ್. RNA ಇಂಟರ್ಫೆರೆನ್ಸ್ ಅಂದ್ರೇನು? It is the silencing of mRNA. ಯಾವ್ದ್ರದ್ದು mRNA ಸೈಲೆನ್ಸ್ ಮಾಡೋದು? ನಿಮ್ಯಾಟೋಡ್ನ mRNA ಟ್ರಾನ್ಸ್ಲೇಷನ್ ಆಗ್ಬಾರ್ದು ಅಂತ ಹೇಳಿ ಏನ್ ಮಾಡ್ತಾರೆ, ಒಂದು ಕಾಂಪ್ಲಿಮೆಂಟರಿ CDNA ಅದನ್ನ ಕ್ರಿಯೇಟ್ ಮಾಡ್ಬಿಟ್ಟು, ಆ ಕಾಂಪ್ಲಿಮೆಂಟರಿ CDNA ಯಾವ ರೀತಿಯಲ್ಲಿ ಕ್ರಿಯೇಟ್ ಮಾಡ್ತಾರೆ ಅಂದ್ರೆ, ಅದರಲ್ಲಿ ಎರಡು DNA ಎರಡು ಸ್ಟ್ರಾಂಡ್ಸ್ಗಳು, ಸೆನ್ಸ್ ಸ್ಟ್ರಾಂಡ್ ಮತ್ತು ಆಂಟಿ ಸೆನ್ಸ್ ಸ್ಟ್ರಾಂಡ್ ಎರಡೂ ಕೂಡ ಟ್ರಾನ್ಸ್ಕ್ರಿಪ್ಷನ್ಗೆ ಒಳಪಡುವ ಹಾಗೆ ಮಾಡ್ತಾರೆ. ಆವಾಗ ಎರಡು mRNA ಪ್ರೊಡ್ಯೂಸ್ ಆಗುತ್ತವೆ. ಅದು ಯಾವುದು ಅದಕ್ಕೆ ಕಾಂಪ್ಯಾಟಿಬಲ್ ಆಗಿರುತ್ತೋ ಆ ಕಾಂಪ್ಯಾಟಿಬಲ್ ಆಗಿರುವಂತ mRNA ಏನ್ ಮಾಡುತ್ತವೆ, ಅದು ಹೋಗಿ ನಿಮ್ಯಾಟೋಡಿನ mRNA ಜೊತೆಗೆ ಬೈಂಡ್ ಆಗಿ ಅಲ್ಲಿ ಡಬಲ್ ಸ್ಟ್ಯಾಂಡರ್ಡ್ mRNA ಪ್ರೊಡ್ಯೂಸ್ ಆಗುತ್ತೆ. ಅವಾಗ ಅದು ಅಲ್ಲಿಗೆ ಅದ್ರ ಕೆಲಸ ಅಂದ್ರೆ ಅಲ್ಲಿ ಮತ್ತೆ ಫರ್ದರ್ ಏನಾಗಲ್ಲ, ಟ್ರಾನ್ಸ್ಲೇಷನ್ ಮಾಡೋದಕ್ಕೆ ಆಗೋದಿಲ್ಲ. ಅದರಲ್ಲಿ ಯಾವುದೇ ಪ್ರೋಟೀನ್ ಅಥವಾ ಎನ್ಸೈಮ್ಸ್ಗಳು ಉತ್ಪತ್ತಿ ಆಗೋದಿಲ್ಲ. ಹಾಗಾಗಿ ನ್ಯೂಟ್ರಿಷನ್ಸ್ ಅನ್ನ ಏನ್ ತಗೊಂಡ್ರು ಕೂಡ ಅದು ಜೀರ್ಣ ಮಾಡ್ಲಿಕ್ಕೆ ಆಗೋದಿಲ್ಲ. ಹಾಗಾಗಿ ಅದು ಸ್ಟಾರ್ವೇಷನ್ ಇಂದಾಗಿ ನಿಮ್ಯಾಟೋಡ್ ಏನಾಗುತ್ತೆ, ಒಳಗಡೆನೆ ಸಾಯುತ್ತವೆ. ಸೊ ಈ ಒಂದು ಇದನ್ನ ಕಾನ್ಸೆಪ್ಟ್ ಅದು ಫಿನಾಮಿನನ್ ಏನ್ ಹೇಳ್ತಾರೆ, RNA ಇಂಟರ್ಫಿರೆನ್ಸ್ ಅಂತ ಹೇಳ್ತಾರೆ, ಅಥವಾ ಸೈಲೆನ್ಸಿಂಗ್ ಆಫ್ RNA ಅಂತ ಹೇಳ್ತಾರೆ. ಸೊ ಈ RNA ಇಂಟರ್ಫಿರೆನ್ಸ್, RNA interference actually it is a natural phenomenon of a defense mechanism. It is present in all eukaryotic organisms. ಎಲ್ಲಾ eukaryotic organisms ನಲ್ಲಿ ಈ RNA interference ಎನ್ನುವಂತ ಒಂದು ಫಿನಮಿನನ್ ನಡೀತಾನೆ ಇರುತ್ತದೆ. ಆದ್ರೆ ಕೆಲವೊಂದು ಆರ್ಗಾನಿಸಮ್ಸ್ ಗಳನ್ನ ಅದು ಏನಾಗೋದಿಲ್ಲ, ಪರಿಣಾಮಕಾರಿಯಾಗಿರಲ್ಲ, ಎಫೆಕ್ಟಿವ್ ಆಗಿರೋದಿಲ್ಲ. ಹಾಗಾಗಿ ಅದರಲ್ಲಿ ಇದು ಒಂದು ಯಾವುದು, ಟೊಬ್ಯಾಕೋ. ಸೊ ಇಟ್ ಈಸ್, ಹಾಗಾಗಿ ಇಲ್ಲಿ ಯಾವ ಕಾನ್ಸೆಪ್ಟ್ ಅನ್ನು ಯೂಸ್ ಮಾಡ್ತಾರೆ, ಇದನ್ನ ಪೆಸ್ಟ್ ರೆಸಿಸ್ಟೆಂಟ್ ಅಂದ್ರೆ ಮೆಲೋಡೋಗೈನೆ ಇನ್ಕಾಗ್ನೇಷಿಯಾ ಎನ್ನುವ ಪೆಸ್ಟ್, ನಿಮ್ಯಾಟೋಡ್ ಪೆಸ್ಟ್, ಅದನ್ನ ಇನ್ಫೆಸ್ಟ್ ಮಾಡದಾಗೆ RNA ಇಂಟರ್ಫಿರೆನ್ಸ್ ಅಥವಾ ಸೈಲೆನ್ಸಿಂಗ್ ಆಫ್ mRNA ಯೂಸ್ ಮಾಡ್ತಾರೆ. ಇದನ್ನ ಏನ್ ಹೇಳ್ತಾರೆ, ನಾಕ್ಔಟ್ ಆಫ್ mRNA ಅಂತ ಹೇಳ್ತಾರೆ, ನಾಕ್ಔಟ್ ಆಫ್ mRNA. ಇಲ್ಲಿ ಡಬಲ್ ಸ್ಟ್ಯಾಂಡರ್ಡ್ ಏನ್ ಮಾಡ್ಬೇಕು, RNA ಪ್ರೊಡ್ಯೂಸ್ ಮಾಡ್ಬೇಕು. ಡಬಲ್ ಸ್ಟ್ಯಾಂಡರ್ಡ್ RNA ಪ್ರೊಡ್ಯೂಸ್ ಮಾಡ್ಬೇಕಂದ್ರೆ ಅದಕ್ಕೆ ಕಾಂಪ್ಲಿಮೆಂಟರಿ DNA ಐಸೋಲೇಟ್ ಮಾಡ್ಬೇಕು. ಕಾಂಪ್ಲಿಮೆಂಟರಿ DNA ಐಸೋಲೇಟ್ ಮಾಡಿ ಅದನ್ನ ಮ್ಯಾನಿಪುಲೇಟ್ ಮಾಡಿ ಅದರ ಎರಡು ಸ್ಟ್ರಾಂಡ್ಸ್ ಗಳು ಡಬಲ್ ಸ್ಟ್ಯಾಂಡರ್ಡ್ mRNA ಪ್ರೊಡ್ಯೂಸ್ ಮಾಡುವಾಗ ನೋಡ್ಕೋಬೇಕು. ಅದನ್ನ ಎಲ್ಲಿ ಅದನ್ನ ಪ್ಲಾಂಟ್ ಸೆಲ್ಸ್ ಗಳಿಗೆ ಯಾವುದು ಡಬಲ್ ಸ್ಟ್ಯಾಂಡರ್ಡ್ ದಿಸ್ ಒನ್ RNA ಏನ್ ಮಾಡ್ತಾರೆ, ಇನ್ಫ್ಯೂಸ್ ಮಾಡ್ತಾರೆ. ಓಕೆ, ಆರ್ CDNA ಡೈರೆಕ್ಟ್ ಆಗಿ ಏನ್ ಮಾಡ್ತಾರೆ, ಸೆಲ್ಸ್ ಗಳಿಗೆ ಇನ್ಫ್ಯೂಸ್ ಮಾಡ್ತಾರೆ. ಓಕೆ, CDNA ಇನ್ಫ್ಯೂಸ್ ಮಾಡಿದ್ರೆ ಇಟ್ ಪ್ರೊಡ್ಯೂಸಸ್ both RNA, and that means two RNAs in both the strands, sense strand as well as anti-strand strands ಗಳು. So what are the sources for the actual sources for this double stranded RNA? ಡಬಲ್ ಸ್ಟ್ಯಾಂಡರ್ಡ್ RNA ನ್ಯಾಚುರಲಿ ಆಗಿ ಒಂದು ಸೆಲ್ಸಿನ ಒಳಗಡೆ ಬರ್ಬೇಕಂದ್ರೆ ಏನಾಗಬೇಕು, ದಟ್ ಶುಡ್ ಬಿ ಇನ್ಫೆಕ್ಷನ್ ಆಗಬೇಕು, ಇನ್ಫೆಸ್ಟೆಡ್ ವಿತ್ ದ ವೈರಲ್ RNA ಜೀನೋಮ್. ಅದರಲ್ಲಿ RNA ಇರ್ಬೇಕು. ವೈರಸ್ ಇನ್ಫೆಸ್ಟೇಷನ್ ಆಗಿ ಅಥವಾ ಇನ್ಫೆಕ್ಷನ್ ಆಗಿ ಅದರಲ್ಲಿ RNA ಇರ್ಬೇಕು. ಇಲ್ಲ ಅದರಲ್ಲಿ ಮೊಬೈಲ್ ಜೀನ್ಸ್ಗಳು, ಜೆನೆಟಿಕ್ ಎಲಿಮೆಂಟ್ಸ್ ಗಳಿರ್ಬೇಕು, ಜಂಪಿಂಗ್ ಜೀನ್ಸ್ ಅಂತೆಲ್ಲ ಹೇಳ್ತಾರೆ, ಟ್ರಾನ್ಸ್ಪೋನ್ಸ್ ಗಳಿರ್ಬೇಕು. ಸೊ ದೀಸ್ ಆರ್ ದಿ ಟೂ ಮೈನ್ ಸೋರ್ಸಸ್ ಫಾರ್ ವಾಟ್ ದ double stranded RNA. Okay, otherwise we have to manipulate it. Okay, so manipulate ಮಾಡಿ ಅದನ್ನ ಇಂಜೆಕ್ಟ್ ಮಾಡ್ಬೇಕಂದ್ರೆ we need to use retroviruses only. We need to use retroviruses only. So here it is given the source of this complementary RNA could be from an infection by viruses having RNA genomes or mobile genetic elements. They are also called transposon. So that replicate via an RNA intermediate. RNA intermediate. So using agrobacterium, in most of the plant biotechnology, the agrobacterium tumefaciens is itself is used as a cloning vector. And if you want to you can also use the retroviruses, the viruses that are having RNAs also you can use. So agrobacterium vectors, the nematode specific genes responsible for parasitic effects were introduced into the host plants. The introduction of DNA was such that it produced both sense and antisense RNA in the host cells. These two RNAs being complementary to each other formed a double-stranded RNA that initiated RNA interference and thus silence the specific mRNA of the nematode. The consequence was that the parasite could not survive in the transgenic. ಹಾಗಾಗಿ ಪ್ಯಾರಾಸೈಟ್ ಈ ನೆಮಟೋಡ್ ಇದೆಯಲ್ಲ ಅದು that roots ನಲ್ಲಿ ಅದು ಬದುಕೋಕೆ ಸಾಧ್ಯನೇ ಆಗಲ್ಲ. Then thereby the plants is protected from the infection. So this is the the image of the root system of the tobacco plant. When it is healthy, it looks like this. When it is infested with the nematode, it see the roots, they are all with the knots. So these are some examples for what the transgenic plants, that means genetically modified crops. Next one, the biotechnological application in medicine. Biotechnological application in medicine. Biotechnological applications in medicine. Diabetes mellitus, what we call it sugar disease, it's the most common disease since all over the world. It's growing very faster. So the remedy is only the insulin injection. But sufficient amount of insulin is not produced in a conventional method. In an older method, the insulin is extracted from the pancreas of the slaughtered animals like cattle, goats, sheep, buffaloes, like that. But it was allergic to the patient. Some of the patients, they get the side effects from this. So to avoid all these problems, these biotechnological scientists, what they developed insulin using the techniques, rDNA technology, rDNA technology. So insulin production is the major application in medicine. Insulin production is the major application in medicine. Insulin is a peptide hormone. It is a peptide hormone. It is usually secreted from the beta cells of the pancreas, islets, beta cells of the islets of pancreas. The insulin hormone is always secreted in its precursor form. In its precursor form, that is called proinsulin. It is inactive. Proinsulin is a inactive form. And this proinsulin, it is made up of three peptide chain. It is made up of three peptide chain. A chain with the 21 amino acid, B peptide chain with the 30 amino acid, and C peptide chain, it is made up of 35 amino acid. So this is A peptide chain, B peptide chain, and C chain or C peptide chain. So non-functional or precursor insulin is having three peptide chains. After processing, processing means when the C peptide chain is removed and it is linked by using the disulfide bonds, it becomes what? Functional insulin. It becomes functional insulin. The functional insulin contains only two chains, A chain and B chain. and B chain. By disulfide bonds, when you treat the proinsulin or precursor of insulin or inactive insulin with any sulfur compound, then what happens? It converts into the functional insulin. Functional insulin. So this is the functional structure of the functional insulin. This is the A chain and B chain. The insulin production in a large the insulin production in a large scale was done by a company in 1983, that was the Eli Lilly company. Eli Lilly company. So this Eli Lilly company, they started producing this insulin using the rDNA technology. rDNA technology. How? So the gene for this A peptide chain and B peptide chain are isolated and they are inserted into the plasmid and rDNA is created, and this rDNA is cloned in a bioreactor, and in a bioreactor the insulin is produced, and then through the downstreaming process the insulin is extracted and it was what formulated in a bottles or in a tubes and all, and then marketed. So this functional insulin, there are this is the method how the insulin is produced using rDNA technology. So the gene from the human pancreatic cell, particularly from the beta cells of the pancreas, is isolated. It is removed and then it is joined using the plasmid, and rDNA is created, and then it is cloned in a bioreactor, and this in the bioreactor the insulin was produced in large quantities and they are purified and they are marketed in a pharmaceutical companies. So in this way the insulin is produced using biotechnology. So insulin is secreted by the beta cells of islets of Langerhans of the pancreas, which controls the amount of glucose in blood. Insulin is a hormone that controls the blood sugar level. Adult onset of diabetes is possible to manage by taking insulin. Insulin used for diabetes was earlier extracted from pancreas of slaughtered cattle and pigs. Insulin from an animal source, though, caused some patients to develop allergy or other types of reactions to the foreign protein. Due to this problem, the idea of synthesizing the insulin by using a bacterium arose. So this is the structure of non-functional or precursor, or it is also called proinsulin, and this is the functional insulin after the removal of the C peptide. And this is the insulin that is produced by any pharmaceutical company, that is called humulin. It is called humulin. Humulin is nothing but human insulin. Humulin means human insulin. In mammals, including humans, insulin is synthesized as a prohormone, which contains an extra stretch called C-peptide. This C-peptide is not present in mature insulin and is removed during maturation into insulin. The main challenge for production of insulin using rDNA technology was getting insulin assembled into a mature form. In 1983, Eli Lilly, an American company, prepared two DNA sequences corresponding to A and B chains of human insulin and introduced them in plasmid PBR322 or PUC18 to transform E. coli to produce insulin chain. So there are two methods of insulin production. Either you remove or separate A gene for A peptide insulin gene. That means insulin A gene as well as insulin gene B gene, and you can clone it in a vector E. coli separately in its two separate bioreactor. Otherwise, you can also extract the entire what insulin gene and you use one what plasmid to clone this the insulin hormone in a bioreactor. So there is two way of production of ಇನ್ಸುಲಿನ್ ನೀವು ಸಪರೇಟ್ ಎರಡು ಜೀನನ್ನೇ ಸಪರೇಟ್ ಮಾಡಿಬಿಟ್ಟು ಸಪರೇಟ್ ಆಗಿ ಕಲ್ಚರ್ ಮಾಡಿ ಅದನ್ನ ಮತ್ತೆ ಆಮೇಲೆ ಅದನ್ನ ಎ ಜೈನ್ ಮತ್ತೆ ಬಿ ಚೈನ್ ಅನ್ನ ಮತ್ತೆ ಅದನ್ನ ಫ್ಯೂಸ್ ಮಾಡಿ ಫಂಕ್ಷನಲ್ ಇನ್ಸುಲಿನ್ ಅನ್ನು ಪ್ರೊಡ್ಯೂಸ್ ಮಾಡಬಹುದು. ಇಲ್ಲ ಒಂದೇ ಇನ್ಸುಲಿನ್ ಜೀನ್ ಅನ್ನ ತಗೊಂಡ್ಬಿಟ್ಟು ಅದನ್ನ ಪ್ಲಾಸ್ಮಿಡ್ಗೆ ಹಾಕಿ RDNA ಪ್ರೊಡ್ಯೂಸ್ ಮಾಡಿ ಅದನ್ನ ಈ ಕೊಲೆನ ಒಳಗಡೆ ಇನ್ಕಾರ್ಪೊರೇಟ್ ಮಾಡಿ ಅದನ್ನ ಬಯೋರಿಯಾಕ್ಟರ್ ಕಲ್ಚರ್ ಮಾಡಿ ಅಟ್ ಎ ಟೈಮ್ ನೀವು ಫಂಕ್ಷನಲ್ ಇನ್ಸುಲಿನ್ ಅನ್ನ ಏನ್ ಮಾಡಬಹುದು ಪ್ರೊಡ್ಯೂಸ್ ಮಾಡಬಹುದು. ಈ ತರ ಎರಡು ಮೆಥಡ್ ಇದೆ. ಸೊ ದಟ್ ಈಸ್ ಅಬೌಟ್ ವಾಟ್ ದ ಇನ್ಸುಲಿನ್ ಪ್ರೊಡಕ್ಷನ್ ಥ್ರೂ ಬಯೋಟೆಕ್ನಾಲಜಿಕಲ್ ಮೆಥಡ್. I hope you all understand this chapter. This next one, that is the biotechnological application in gene therapy. So gene therapy. So you know that so many people are suffering from genetic disorders with a defective gene. So when these disorders are not able to cure through the conventional medicines, antibiotics, even if you give antibiotics, vaccines, these diseases can be not cured. So what you have to do is to these genetic disorders itself is caused by the defective gene. By birth, you will have that gene, defective gene. That is why that faulty gene you are already having, so you cannot, even if you take the medicines, you cannot cure it. So for this problem, what you have need, what is the needed thing? That is the incorporation of or replacement of the correct, the healthy genes into the body cells, So that is the gene therapy. Another biotechnological application in medicine is gene therapy. Gene therapy is the therapeutic use of biotechnology. Therapeutic use of biotechnology. That is the method of replacement of defective gene by healthy functional gene. By healthy functional gene. The gene therapy was first used in 2-year girl child who was suffering from ADA deficiency disease or ADA deficiency disorder. ADA deficiency disorder. This patient is born with the defective gene, ADA gene, defective ADA gene. Because of this defective ADA gene, adenosine deaminase enzyme adenosine deaminase production is not possible. So adenosine deaminase enzyme is not produced in that patient's body. So because of the adenosine deaminase is absent, the adenosine is not metabolized properly. Adenosine is not metabolized properly. So this affects the T lymphocytes. This affects the production of the T lymphocytes. Affects the production of T lymphocytes. That is why that patient that suffers from the immune deficiency, suffer from the immune deficiency. That means either the patient does not produce any antibodies, or even if the antibodies are produced, they are produced in a very, very small quantities due to the absence of the T lymphocytes. So this deaminase has to metabolize properly for the production of the normal T lymphocytes, for the production of normal T lymphocytes. So ADA deficiency disorder anna ಸಿವಿಯರ್ ಕಂಬೈನ್ಡ್ ಇಮ್ಯುನೋಡಿಫಿಷಿಯನ್ಸಿ ಸಿಂಡ್ರೋಮ್ ಅಂತ ಹೇಳ್ತಾರೆ. ಸೊ ಕಾಯಿಲೆಗಳನ್ನ ತಡೆಗಟ್ಟುವಂತ ಸಾಮರ್ಥ್ಯ ಇವರಲ್ಲಿ ಇರೋದಿಲ್ಲ. ಯಾಕೆ ಇರಲ್ಲ ಅಂತಂದ್ರೆ ಅವರಲ್ಲಿ ಹುಟ್ಟುವಾಗ್ಲೇನೆ ಜೀನ್ ಇರೋದಿಲ್ಲ, ಅಥವಾ ಜೀನ್ ಅಬ್ಸೆಂಟ್ ಆಗಿರುತ್ತೆ, ಇಲ್ಲ ಜೀನ್ ಇದ್ದಿದ್ದೇನಾಗಿರುತ್ತೆ, ಫಾಲ್ಟಿ ಆಗಿರುತ್ತೆ, ಡಿಫೆಕ್ಟಿವ್ ಆಗಿರುತ್ತೆ. ಅಂತ ಸಂದರ್ಭದಲ್ಲಿ ಆ DNA ಆ ಜೀನ್ ಯಾವ್ದನ್ನ ಆ ಎನ್ಸೈಮ್, ಒಂದು ಪರ್ಟಿಕ್ಯುಲರ್ ಎನ್ಸೈಮ್ ಇದೆ, ಒಂದು ಸ್ಪೆಸಿಫಿಕ್ ಎನ್ಸೈಮ್ ಇದೆ, ಎಡಿನೋಸಿನ್ ಎನ್ನುವಂತ ನ್ಯೂಕ್ಲಿಯೋಸೈಡ್ ಅನ್ನ ಡೈಜೆಸ್ಟ್ ಮಾಡ್ಲಿಕ್ಕೆ, ಮೆಟಬಲೈಸ್ ಮಾಡ್ಲಿಕ್ಕೆ ಒಂದು ಪರ್ಟಿಕ್ಯುಲರ್ ಎನ್ಸೈಮ್ ಇದೆ. ಅದರ ಹೆಸರು ಎಡಿನೋಸಿನ್ ಡಿಅಮಿನೇಸ್ ಎನ್ಸೈಮ್. ಅದು ಉತ್ಪತ್ತಿ ಆಗೋದಿಲ್ಲ, ಸರಿಯಾದ ಪ್ರಮಾಣದಲ್ಲಿ ಅದು ಪ್ರೊಡಕ್ಷನ್ ಆಗೋದಿಲ್ಲ. ಅದು ಪ್ರೊಡಕ್ಷನ್ ಆಗಿಲ್ಲ ಅಂದ್ರೆ ಅದು ಎಡಿನೋಸಿನ್ ಏನಾಗುತ್ತೆ, ಬಾಡಿಯಲ್ಲಿ, ಬಾಡಿ ಫ್ಲೂಯಿಡ್ ನಲ್ಲಿ ಅದು ಅಕ್ಯುಮುಲೇಟ್ ಆಗ್ತಾ ಹೋಗುತ್ತೆ. ಆವಾಗ ಮಕ್ಕಳಲ್ಲಿ, ಚಿಕ್ಕ ಮಗುವಿನಲ್ಲಿ ಏನಾಗುತ್ತೆ ಅಂದ್ರೆ ಸರಿಯಾಗಿ ಟಿ ಲಿಂಫೋಸೈಟ್ಸ್ ಗಳು ಥೈಮಸ್ ಗ್ಲ್ಯಾಂಡ್ ನಲ್ಲಿ ಪ್ರೊಡ್ಯೂಸ್ ಆಗೋದಿಲ್ಲ. ಹಾಗಾಗಿ ಅವರಿಗೆ ತೀವ್ರವಾದಂತಹ ಏನು ಕಾಯಿಲೆಗಳು ಬರುತ್ತೆ. ಅಂದ್ರೆ ಯಾವುದೇ ರೋಗವನ್ನು ತಡೆಗಟ್ಟುವಂತಹ ಸಾಮರ್ಥ್ಯ ಇರುವುದಿಲ್ಲ, ರೋಗ ಪ್ರತಿರೋಧಕ ಶಕ್ತಿಯನ್ನ ಕಳ್ಕೊಂಡಿರ್ತಾರೆ. ಅದಕ್ಕೆ ಆ ಕಾಯಿಲೆಗೆ ಏನ್ ಹೇಳ್ತಾರೆ, ADA deficiency disorder, or it is also called severe combined immunodeficiency diseases. Diseases as well as disorders. So there were so many methods they used to treat these diseases. They used what? The bone marrow transplantation. ಬೋನ್ ಮ್ಯಾರೋ ಟ್ರಾನ್ಸ್ಪ್ಲಾಂಟೇಶನ್ ಅನ್ನ ಕೂಡ ಮಾಡಿ ನೋಡಿ. ಬೋನ್ ಮ್ಯಾರೋ ಟ್ರಾನ್ಸ್ಪ್ಲಾಂಟೇಶನ್ ಅಲ್ಲಿ ಏನ್ ಮಾಡ್ಬೇಕು, ಬೋನ್ ಮ್ಯಾರೋನೇ ಕಸಿ ಮಾಡ್ಬೇಕು. ಹೊಸ ಹೊಸ ಬೋನ್ ಮ್ಯಾರೋನ ಪದೇ ಪದೇ ಹಾಕ್ತಾ ಇದ್ರೆ ಅವಾಗ ಅದನ್ನ ಕಂಟ್ರೋಲಿಗೆ ತರಬಹುದು. ಯು ಕೆನಾಟ್ ಪ್ರಿವೆಂಟ್ ಇಟ್. ಓಕೆ. ಇನ್ನೊಂದು ಮೆಥಡ್ ಎನ್ಸೈಮ್ ರಿಪ್ಲೇಸ್ಮೆಂಟ್. ಈ ಎನ್ಸೈಮ್ ಅನ್ನ ಪದೇ ಪದೇ ಕೊಡ್ತಾ ಹೋಗ್ಬೇಕಾಗುತ್ತೆ. ಯಾಕಂದ್ರೆ ಎಡಿನೋಸಿನ್ ಅನ್ನ ಮೆಟಬಲೈಸ್ ಮಾಡ್ಲಿಕ್ಕೆ ಎನ್ಸೈಮ್ ಅನ್ನ ಅವಾಗ ಕೊಡ್ತಾ ಹೋಗ್ಬೇಕಾಗುತ್ತೆ. ಅದು ಕೂಡ ಅಷ್ಟೊಂದು ಇಫೆಕ್ಟಿವ್ ಆಗಲ್ಲ. ಸೊ ಹಾಗಾಗಿ ಕೊನೆಗೆ ಇನ್ನೊಂದು ಏನು, ಕೊನೆಯದು ಒಂದು ರೆಮಿಡಿ ಅಂದ್ರೆ ಜೀನ್ ಥೆರಪಿ. ಕೊನೆ ಜೀನ್ ಥೆರಪಿಯಲ್ಲಿ ಏನ್ ಮಾಡ್ಬೇಕಾಗುತ್ತೆ ಅಂದ್ರೆ ಟಿ ಲಿಂಫೋಸೈಟ್ ಅನ್ನ ಪೇಷಂಟ್ ಬಾಡಿಯಿಂದ ತೆಗೆದ್ಬಿಟ್ಟು ಅದಕ್ಕೆ ಹೆಲ್ದಿ ಜೀನ್ ಅನ್ನ ರಿಟ್ರೋವೈರಸ್ ಅನ್ನ ಬಳಸಿ ಏನ್ ಮಾಡ್ಬೇಕು, ಇನ್ಕಾರ್ಪೊರೇಟ್ ಮಾಡ್ಬೇಕು. ಆಮೇಲೆ ಅದನ್ನ ಪೆಟ್ರಿಡಿಷನಲ್ ಮಾಡಿ, ಫಸ್ಟ್ ಗೆ ಬ್ಲಡ್ ಸ್ಯಾಂಪಲ್ ಅನ್ನ ಕಲೆಕ್ಟ್ ಮಾಡ್ಕೋಬೇಕು, ಅದನ್ನ ಟಿ ಲಿಂಫೋಸೈಟ್ ಅನ್ನ ಐಸೋಲೇಟ್ ಮಾಡ್ಬೇಕು, ಟಿ ಲಿಂಫೋಸೈಟ್ಸ್ ಅನ್ನ ಪೆಟ್ರಿಡಿಷನಲಿ ಕಲ್ಚರ್ ಮಾಡ್ಬೇಕು. ಕಲ್ಚರ್ ಮಾಡಿದ ಟಿ ಲಿಂಫೋಸೈಟ್ಸ್ ಗಳಿಗೆ ರಿಟ್ರೋವೈರಸ್ ಅನ್ನ ಯೂಸ್ ಮಾಡ್ಕೊಂಡು, ವೆಕ್ಟರ್ ಆಗಿ ಯೂಸ್ ಮಾಡ್ಕೊಂಡು ಅದಕ್ಕೆ ಯಾವ್ದು ಹೆಲ್ದಿ ಜೀನ್ ಅನ್ನ, ADA ಜೀನ್ ಅನ್ನ ಹಾಕ್ಬೇಕು. ಮತ್ತೆ ಮರಳಿ ಇನ್ಫ್ಯೂಸ್ ಮಾಡ್ಬೇಕು. ದೆನ್ ಅಗೇನ್ the T lymphocytes from the culture is infused back to the blood of the patient. ಈ ತರ ಮಾಡ್ಬೇಕಾಗುತ್ತೆ. ಬಟ್ ಸ್ಟಿಲ್ ಇದು ಇಫೆಕ್ಟಿವ್? ನೋ, ಇಟ್ ಇಸ್ ನಾಟ್ ಇಫೆಕ್ಟಿವ್. ವೈ? Because these T lymphocytes are not immortal. They are not immortal. They have a lifespan. ಅದಕ್ಕೂ ಕೂಡ ರೆಡ್ ಬ್ಲಡ್ ಸೆಲ್ಸ್ ತರನೇ ಒಂದು ಸರ್ಟನ್ ಲಿಮಿಟೆಡ್ ಪೀರಿಯಡ್ ವರೆಗೆ ಮಾತ್ರ ಬದುಕುತ್ತೆ. ಹಾಗಾಗಿ ಇದನ್ನು ಕೂಡ ಪೀರಿಯಾಡಿಕಲಿ ಮಾಡ್ತಾ ಹೋಗ್ಬೇಕಾಗುತ್ತೆ. ಹಾಗಾಗಿ ಇದನ್ನು ತಡೆಗಟ್ಟಲಿಕ್ಕೆ ಏನ್ ಮಾಡ್ಬೇಕು, ಪರ್ಮನೆಂಟ್ ಕ್ಯೂರ್ ಮಾಡ್ಬೇಕಂದ್ರೆ ಎಂಬ್ರಿಯಾನಿಕ್ ಸ್ಟೇಜ್ ನಲ್ಲಿ ಇದ್ದಾಗ್ಲೇನೆ, ಅಂದ್ರೆ ಎಂಬ್ರಿಯಾನಿಕ್ ಸ್ಟೇಜ್ ಯು ಆಲ್ ನೋ, ಡ್ಯೂರಿಂಗ್ ದ ಬ್ಲಾಸ್ಟುಲರ್ ಸ್ಟೇಜ್ ನಲ್ಲ, ಗ್ಯಾಸ್ಟ್ರುಲರ್, ಇದರಲ್ಲಿ ಏನ್ ಮಾಡ್ಬೇಕು, ಸೆಲ್ಸ್ ಗಳಿಗೆ ಜೀನ್ ಅನ್ನ ಹಾಕ್ಬಿಟ್ಟಾಗ ವೆನ್ ದ ಎಂಬ್ರಿಯೋ ಡೆವಲಪ್ಸ್ ಇನ್ ಟು ಹೋಲ್ ಆರ್ಗಾನಿಸಮ್ಸ್, ಒಂದು ಫೀಟಸ್ ಕಂಪ್ಲೀಟ್ ಡೆವಲಪ್ ಆದ್ಮೇಲೆ ಏನಾಗುತ್ತೆ, ಆ ಎಲ್ಲಾ ಸೆಲ್ಸ್ ಗಳಿಗೂ ಕರೆಕ್ಟ್ ಆಗಿರುವಂತ ಜೀನ್ ಬರುತ್ತೆ. ಆವಾಗ ಇದನ್ನ ಪರ್ಮನೆಂಟ್ ಕ್ಯೂರ್ ಮಾಡ್ಕೋಬಹುದು. ಇಲ್ಲದಿದ್ರೆ ಇದು ಈ 3 ಮೆಥಡ್ ನಲ್ಲಿ ಕೂಡ ಇದು ಏನ್ ಕೊಡೋದಿಲ್ಲ, ಪರ್ಮನೆಂಟ್ ಆಗಿ ಪರ್ಮನೆಂಟ್ ಟ್ರೀಟ್ಮೆಂಟ್ ಅನ್ನು ಕೊಡೋದಿಲ್ಲ ಅಥವಾ ಪರ್ಮನೆಂಟ್ಲಿ ಇಫೆಕ್ಟಿವ್ ಆಗಿರಲ್ಲ. ಓಕೆ, ವಾಟ್ ಆರ್ ದಿ ಮೆಥಡ್ಸ್ used to treat this ADA deficiency disease? That is, one is the bone marrow transplantation, the second one is enzyme replacement therapy, the third one is the gene therapy using the T lymphocytes, and the fourth one, that is the gene therapy itself, that is using the that is the modified or the functional gene which was incorporated or introduced during the embryonic stage. So the insertion or introduction of the healthy functional gene into the cells during its embryonic stage is the only remedy for the disease, ADA deficiency diseases. So today, this much we have discussed. I hope you understood. I think so. In the next class, we'll discuss about the questions regarding this chapter. So thank you.